Hello everyone. I am going to discuss Chapter 15, Lesson 3, the applications of genetic engineering, particularly on treating diseases and personal identification. This is found in your textbook pages 431 to 434. Our objectives: explain how recombinant DNA technology improves human health. We're also going to summarize the process of DNA fingerprinting and explain its uses. So let's start with one of the applications of genetic engineering. Again, we've mentioned one of which is treating diseases. A process called gene therapy is used, in which they change the gene to treat a medical disease or disorder. In gene therapy, an absent or faulty gene is replaced by a normal working gene. This process allows the body to make a protein or the protein or enzyme it needs, which eliminates the cause of the disorder. This is an example of gene therapy. To deliver therapeutic genes to target cells, researchers engineer a virus that cannot reproduce or cause harm. This is the normal gene, normal hemoglobin gene, and this is the genetically engineered virus. Then the DNA containing the therapeutic gene is inserted into the modified virus. This is now the inserted normal gene into the DNA of the virus. The patient's cells are then infected with the genetically engineered virus. Here is a virus. It has been inserted its DNA into the patient's cell. In theory, supposedly the virus will insert the healthy gene into the target cell and correct the defect. But gene therapy can be risky. Why is that so? In 1999, 18-year-old Jesse Gelsinger volunteered for a gene therapy experiment designed to treat a genetic disorder of his liver. Unfortunately, he suffered a massive reaction from the viruses used to carry genes into his liver cells, and he died a few days later. So for gene therapy to become an accepted treatment, we need more reliable ways to insert working genes and to ensure that the DNA used in the therapy does no harm. So another ways of how genetic engineering can improve human health is through genetic testing. This can be used to determine if two prospective parents are carrying the alleles for a genetic disorder, let's say for example cystic fibrosis or CF. Because the CF allele has slightly different DNA sequences from its normal counterpart, genetic tests use labeled DNA probes that can detect and distinguish the complementary base sequences found in the disease-causing alleles. Some genetic tests search for changes in cutting sites of restriction enzymes, while others use PCR or polymerase chain reaction to detect differences between the lengths of normal and abnormal alleles. Nowadays, genetic tests are available for diagnosing hundreds of disorders. We have mentioned this before in our previous lessons, example the Turner syndrome, Huntington's disease, Down syndrome, and the like. Another way in which genetic engineering is helpful in treating diseases is through examining active genes. In this way, scientists are able to distinguish between normal cells and cancer cells. The same genes are not active in every cell. By studying which genes are active and which are inactive in different cells, scientists can understand how the cells function normally and what happens when genes don't work as they should. Scientists use DNA microarray technology to study hundreds or even thousands of genes at once to start to understand their activity levels. So what is DNA microarray? A DNA microarray is a glass slide or silicon chip to which spots of single-stranded DNA has been tightly attached. Typically, each spot contains a different DNA fragment. Differently colored tags are used to label the source of DNA. So for example, to compare the genes in cancer cells with genes in normal cells, first we're going to prepare the cDNA probe, and the mRNA samples are isolated from two different types of cells or tissues, such as the cancer cells and the normal cells. Then the enzymes are used to prepare complementary DNA molecules, or cDNA, from both groups of mRNA. Contrasting fluorescent labels are attached to both groups of cDNA, the red to one or green to the other. Then microarrays are prepared. The DNA fragments corresponding to different genes are bound to the wells in microarray plate. The single strand of DNA are attached to the wells in the plate. So this is an enlarged image of the wells in the plate. Then combining the probe and microarray samples, labeled complementary DNA molecules bind to complementary sequences on the plate. Here we have the labeled cDNA molecules. The red ones are for the cancer cells and the green ones are for the normal cells. Then both samples of labeled DNA would be mixed together and they would then complete to bind the complementary DNA sequences already in the microarray. If the cancer cell produces more of a particular form of mRNA, then more red labeled molecules will bind at the spot for that gene, turning it red. Where the normal cell produces more mRNA for another gene, the spot will be green. Where there is no difference between the two cell types, the spot will be yellow because it contains both colors. Another application of genetic engineering is to identify individuals or personal identification. This helps in solving crimes and as well as establishing family relationships. So how is DNA used to identify individuals? DNA fingerprinting analyzes sections of DNA that may have little or no function but that vary widely from one individual to another. No individual is exactly like any other genetically, except for identical twins who share the same genome. Chromosomes contain many regions with repeated DNA sequences that do not code for proteins. These vary from person to person. Here, one sample has 12 repeats between genes A and B, while the second has 9 repeats between the DNA genes. The DNA fingerprinting can be used to identify individuals by analyzing the sections of DNA that may have little or no function, but that vary widely from one individual to another. So how is DNA fingerprinting done? First, restriction enzymes cut a sample or a small sample of human DNA into fragments containing genes and repeats. Note that repeat fragments from these two samples are of different lengths. So different repetitions, different lengths. Next, gel electrophoresis separates the restriction fragment by sizes by inducing voltage in both ends of the gel or plate. A DNA probe detects the fragments that have highly variable regions, revealing a series of variously sized DNA bands. We've learned before that the shorter fragments move faster at the end of this gel plate. The longer bands stays here, separating these fragments into sizes. If enough combinations of enzymes and probes are used, the resulting patterns of bands can be distinguished statistically from that of any other individual in the world. DNA samples can be obtained from blood, sperm, or tissue, even from a hair strand if it has tissue at the root. DNA fingerprinting is also very useful in forensic science. The precision and reliability of DNA fingerprinting has revolutionized forensics. This is a scientific study of crime scene evidence. DNA fingerprinting has helped solve crimes, convict criminals, and even overturn wrongful convictions. To date, DNA evidence has saved more than 110 wrongfully convicted prisoners from death sentences. DNA forensics is used in wildlife conservation as well. Like in African elephants, they are highly vulnerable species. Poachers who slaughter the animals mainly for their precious tusk have reduced their population dramatically. So to stop ivory trade, African officials now use DNA fingerprinting to identify the herds from which black market ivory has been taken. Now, how do we identify our family relationships? When genes are passed from parent to child, genetic recombination scrambles the molecular markers used for DNA fingerprinting, so ancestry can be difficult to trace. The Y chromosome, however, never undergoes crossing over, so the only males carry it. Therefore, Y chromosomes pass directly from father to son with few changes. Similarly, the small DNA molecules found in mitochondria are passed with very few changes from mother to child in the cytoplasm of the egg cell. Because mitochondrial DNA is passed directly from mother to child, your mitochondrial DNA is the same as your mother's, which is also the same as her mother's or your grandmother. This means that if two people have an exact match in their mitochondrial DNA, then there is a very good chance that they share a common maternal ancestor. Now, Y chromosome analysis has helped researchers settle long-standing historical questions. One such question, did President Thomas Jefferson father the child of a slave, may have been answered in 1998. DNA testing showed that descendants of the son of Sally Hemings, a slave on Jefferson's Virginia estate, carried his Y chromosome. This result suggests Jefferson was the child's father, although the Thomas Jefferson Foundation continues to challenge that conclusion. That ends our discussion, so let's now move on to the lesson check questions. So I will be pausing for some time for you to think of your answer after you have given the question. For our first question, name two uses for recombinant DNA technology. Think of your answer. Now here's the answer. Recombinant DNA technology improves health by preventing disease and providing treatment for diseases. It is also used to identify individuals. Moreover, it is also helpful in agriculture and industry. For the second question, describe how a DNA microarray might be used to distinguish normal cells from cancer cells. Think of your answer. The correct answer is DNA microarrays allow scientists to compare the gene expression patterns of different cells. This kind of comparison can be used to distinguish cancer cells from normal cells. Great job! For our next question, describe two important uses for DNA fingerprinting. Take some time to think of your answer. The correct answer is DNA fingerprinting can be used to help solve crimes by matching DNA evidence from a crime scene with possible suspects. It can also be used to establish familial relationships, such as in cases of disputed paternity, by finding alleles in a child that are not carried by the mother and then matching them to the father's fingerprint. So those are our questions, and that ends our discussion. Thank you so much.